I think it doesn't matter to me. I think that's about it. Well, welcome to Prime Time in the Library. I'm Deb Carlos, the Dean of the College, and I'm really excited to invite you to this session as part of the Prime Time in the Library series. And the series is held by the library in order to work with a number of offices and departments around campus who really promote lifetime learning. And so it's always fun to see the mix of students and faculty and staff who come to these because they're really interested in learning what are our colleagues doing and what can we learn from that. So welcome to Prime Time in the Library series. A couple of announcements, and I'm going to read the first one so I get all the details right. All students who are working on a project or paper may want to consider applying for the $250 Library Research Prize. The deadline is February 15th. And to qualify to submit, you need to be not graduating before May 2012, right? Not before May 2012. The work can be in any medium of paper or video. The work must have been completed in the spring or fall 2011 or interim 2012. So last spring, this fall, this interim. And the application doesn't require any additional work. So it's not like you have to do something on top of what you're already doing, beyond what you're submitting for your coursework. You just need to write a short essay about the research process. There's information on the library website, so if you're interested in applying for the Library Research Prize, it's $250. Check that out. Again, it's due February 15th. Today's the last primetime series for the fall semester, so there's certainly a whole new series. Does it start in the spring? Yeah, so we don't do it. Okay, so spring, watch for the whole new series of Prime Time in the Library. So for this session, it's my pleasure to introduce Dr. Joy Doan from the Biology Department, and Ruth Lundeen and Kent Nichols, both senior biology majors, and Kent is also a psych major. And they're going to present their research that was part of the Edwin Scholars Award. And the Edwin Scholars program comes out of my office. It's a competitive process for faculty and student researchers. They apply for the award. They're reviewed by other faculty. And it funds summer research between students and faculty. And it's named after John Alexis Edgren, who was our founder of Bethel, and was really passionate about interaction between students and faculty. So Jay Barnes, our now president, launched this award when he was provost. And it's really appropriate that we call it the Edgren Scholars Award, because Edgren really believed, even in the late 1800s, that one of the most important things of education should be the collaboration between students and faculty. So for their research, I'm going to read you the title of their research. And then I confessed to Joy that I wasn't going to read the summary of their research because I was destined to mispronounce it and not really fully understand it. So the title is Regulation of Cellular and Molecular Macrophage Functions by Lipid Rafts. And I had the chance to ask Joy before we started a little bit about, so how does this connect for a layperson to something that I might understand? How does this really sort of pure basic science research connect to things that we would understand? And he gave me a great answer, so I hope that you tell us that before the end. So welcome.
All right, thank you. Is it okay if I stand over here? All right, thanks. I might move around a little bit too. So thank you, Deb, for the kind introduction. I'm Joy Doan, and I work down in the biology department. And my research interest has been in the area of macrophage activation, essentially since I started graduate school. Today we're going to talk about the work that we did last summer, so summer of 2011, under the Edwin Scholars Program. And this is an overview of where we're going this morning. I'm going to provide the overview to the project. So I'm going to talk a little bit about host immunity, what macrophages are. If you have a little bit of background in Greek and or Latin, you might already be thinking big eaters, and that's a good thing. And we're going to talk about these structures called lipid rafts. Then I'm going to pass off to my student collaborators, Kent, who will talk about the regulation of phagocytosis by these structures, and Luke, who's going to talk about another part of inflammation known as nitric oxide production. And then they're going to hand back off to me, and I'll talk about what I would like to do next summer. So I thought it would be useful, given that this is probably a largely non-science audience, to talk a little bit about where the immune system lives in our bodies. Technically, the immune system is responsible for protecting all of our body's tissues from assault from the outside environment, typically from pathogens or things that make us sick. Immune responses technically begin when our leukocytes or white blood cells develop. They develop in two key parts of the body, the bone marrow, which is highlighted here in the pelvic region, and the long bones of the legs, because that's where it's most active in adults, and in the thymus, which is a small butterfly-shaped gland that sits right on top of your heart. This is where most of your white blood cells do their development. Once the cells have been developed, they're released into your circulatory system, and your heart just pumps them through your body. But in order to protect your tissues, they have to be able to leave the bloodstream. And that's what your lymph nodes are for. You're all probably familiar with lymph nodes because they're the things that get large and painful when you get sick. But your white blood cells actually leave the bloodstream and move through the lymph nodes roughly 2 times every day so that they can sample what's going on in your body tissues. Triggered them. Awesome. And so that does a very good job of largely protecting your circulatory system and your tissues. But in order to bring pathogens or other types of debris that might be harmful to your body in contact with these white blood cells, which typically go from blood to your lymph nodes and back, like let's say you get a cut on your big toe, we have to have some resident cells that live in all of our tissues in order to protect the tissues as a first line of defense. The cell type that I study, called the macrophage, is one of these resident cells, and there are various types of macrophages that are resident in essentially every tissue in your body, and they serve as a major first line of defense. Literally defined, the cell is a big eater, and when it comes into contact with foreign material from outside the body, it consumes it, ingests it, digests it, and then participates in the activation of more advanced functions. So as I mentioned, this is a macrophage. This is a little scanning electron micrograph. The macrophage is shown in yellow, so you can get an idea of its very convoluted three-dimensional surface. And this macrophage is interfacing with some E. coli bacteria. I was not cool enough to take that picture. I borrowed it from the internet. These cells have a variety of different functions that contribute to host defense. Their primordial function, which is what they were named for, is phagocytosis, so literally cell eating. If we could take a look at this macrophage, say 20 minutes from when this picture was taken, what you would see is that many of these bacteria would be inside being actively consumed by the cell for the purposes of activating the immune response. So phagocytosis is one aspect of the antimicrobial defenses of a macrophage. There are a variety of other things that macrophages do that are antimicrobial. These include the production of mediators that contribute to inflammation. So Kent's going to be talking about phagocytosis, and Luke is going to be talking about one of the major markers of inflammation, which we all sort of experience as a collection of symptoms where we have redness, pain, heat, and swelling in the various parts of our body that get inflamed. But the production of a chemical called nitric oxide is a key marker of inflammation. Beyond that, macrophages stick around for the duration of an immune response and contribute to the healing of that wound. And so when we talk about wounds from an immunological perspective, it's not just about cuts or breaks in bones or physical breaches, but we can also consider infection to be a specific kind of wounding that damages tissues and that needs to get repaired in order to restore the body to full function. And then finally, macrophages are an important bridge to what we call adaptive immunity. These would be the more advanced functions of immunity. Without macrophages, they don't happen. But if you take these away, you also have very severe immune deficiencies. So macrophages are kind of involved in the whole spectrum of immune defenses. And if they don't do their jobs on the front end, specifically phagocytosis and inflammation, it's very challenging for the immune system to pick up and carry through with the back end of a response. Now, the model that we use for studying macrophages in the lab is not terribly complicated. It's a model that's been well defined for a number of decades. And it involves taking resting macrophages, which can actually be grown in little plates in culture, and then turning them into activated cells. The resting macrophages that we use in our research come from mouse bone marrow. If you're really interested, we can tell you how we get them later, but if you've had a snack, you might not want me to talk about that right now. Just trust me. So we get these cells from mouse bone marrow, which are actually immature blood cells. We grow them in the presence of chemicals to turn them into resting macrophages, and we get these beautiful homogeneous populations. These cells basically will do a little bit of phagocytosis, and that's about all they'll do. However, if you provide them with a couple of key stimuli, they gain a lot of functional attributes. The classic way for turning a resting macrophage into an activated macrophage is to use the chemicals interferon-gamma and lipopolysaccharide. Interferon-gamma is sometimes called a priming stimulus. in the context of a normal immune response, interferon-gamma is produced by your body to turn the macrophages on. And so it's kind of your body saying, we have sensed an invader, and we would like the macrophages to become active. As a check on the interferon-gamma stimulus, we also need to have a stimulus that comes from an invader itself. A classic activator of macrophages is lipopolysaccharide, which comes from bacteria. So in the presence of a pathogen stimulus and a host stimulus together, that confirms to the macrophage that there is indeed an infection and the cell is allowed to become activated. When it becomes activated, it gains a variety of functions. Its phagocytic response is dramatically enhanced. It engages in what's called a respiratory burst, where it actually uses oxygen to produce toxic chemicals like hypochlorite, which is the active ingredient in bleach, hydrogen peroxide, which we know we can purchase at the grocery store and dump on cuts and it bubbles, and interesting and exciting sounding things like superoxide anion. It sounds insidious, and it kind of is. Right? The respiratory burst, together with a variety of other things, enables the macrophage to induce inflammation, which will then help recruit other cells to the site of infection. And then it engages in stimulation of other white blood cells. And so what we look for what we're looking for is how this stimulation actually happens. So when these stimuli interact with the cell, they can't actually get across the cell's membrane. So I'm going to go back for a second, and you'll notice that one of the defining characteristics from a cell is that it has a boundary. And one of the defining characteristics of that boundary is that it's not terribly permeable to things in its outside environment. So for that reason, when we add stimuli, if they come up against the cell, they really just bump into it. So in order to activate the cell, they need to cause some kind of a change outside the cell that results in a change inside the cell. And so the common model that's used for cell membranes is that of a mosaic or a fluid mosaic. So I put a mosaic that all of you might recognize over here on the left to point out that you can actually create a very functional, beautiful picture out of a series of small little pieces, which if you saw them individually would have very little meaning to you. Now, when we put them all together, we see Van Gogh's Starry Night, or an approximation thereof out of tiles. Cell membranes do essentially the same thing. They're made out of little component parts, but instead of tiles, we have lipids, or we have proteins, or we have carbohydrates, and they all get assembled together into a functional picture. Where this analogy really breaks down, however, is that in an art mosaic, the pieces are generally fixed, whereas in a membrane mosaic, the pieces aren't fixed, and they flow past one another and create movement and this dynamic nature that enables cells to respond. In spite of the fact that there's tons of movement in this membrane, there are small regions within the membrane that are more highly organized and less fluid, and these are called lipid rafts. Lipid rafts, in terms of their composition, have specialized lipids present in them. They contain more cholesterol, which is sort of diagrammed in these orange key-shaped bars here. They have more cholesterol than the rest of the membrane. They also have their lipids have unique properties that make them stick together a little bit more than what you see in the rest of the cell's membrane. And for this reason, lipid rafts are frequently called liquid-ordered structures. In other words, they have more order and less fluidity than the rest of the membrane. Because of this, they can recruit and trap molecules that contribute to cell communication. So when interferon-gamma and lipopolysaccharides bump up against the cell, they're actually bumping up against receptors that live in these lipid rafts. And because those receptors are organized, it enables the macrophage to efficiently recruit and activate what's happening on the inside of the cell. So it makes it really efficient for cells. So the main question that we're then asking, because it's already known that the receptors for both interferon-gamma and lipopolysaccharide live in these lipid rafts, is whether or not the macrophage can still become activated if you disrupt the architecture of lipid rafts. And so the basic model that we're using here is we're using a chemical called methyl beta cyclodextrin that disrupts lipid rafts by simply serving as a cholesterol vacuum. It will vacuum about 4 40% of the cholesterol out of any given cell membrane, and that is more than sufficient to disrupt the architecture of these lipid rafts. That's really well documented in the literature. And so then what we're testing is whether or not disrupting the lipid rafts can also result in a reduction or an alteration in the signaling capacity of the cell and of the cell's function. The specific aims that we had for the Edgren project last summer were to approach both cell signaling and cell function from two directions. And so Kent approached the question of whether or not macrophages with disrupted membrane architecture can still engage in that eating process known as phagocytosis. And Luke approached whether or not disruption of these structures could disrupt inflammatory processes. And so at this point, I'm going to hand off to Kent and let him tell you about what he did with phagocytosis.
Thank you very much. As Dr. Doan said, my portion of the research focused on phagocytosis, or the eating of the macrophages. And for this portion, we had a couple of aims. First off, we wanted to develop a method that we could track the amount of phagocytosed particles. Before, we had never underwent a procedure for phagocytosis assay in the lab. So over the summer, we wanted to develop this method. And to develop this, we wanted to first understand if using an opsonizing agent, which is supposedly supposed to increase the amount of phagocytosed particles or increase the functioning of a macrophage, if that had any statistical significance in changing function of macrophages. And also, we wanted to see how duration, if you allow a macrophage to phagocytose particles, how does the amount of time you give them affect the ability for the macrophages to engulf more particles, basically. Next, we wanted to determine specifically how the interferon-gamma, how that pathway is affected by Raft disruption, and to see if interferon-gamma, that cell signaling pathway, goes through Rafts, basically, or uses the Rafts. And to do this was our experimental approach. So we cultured bone marrow macrophages into three separate conditions, basically. So on the right is the Raft disrupted with a cyclodextrin and with a matched medium. And on the left is normal medium, basically. Then after putting in these conditions, we either stimulated them with interferon-gamma and LPS combined, or we left them unstimulated. And after a 24-hour period of time, we used particles called zymosin, fluorescently labeled zymosin particles, which macrophages are known to phagocytose readily. And this is the portion. After 24 hours, we introduced the zymosin particles into the culture, and then we allowed them to phagocytose the particles, and after that we fixed the cells and fluorescently labeled them, and then we analyzed them using fluorescent microscopy. So, since we ran a few trials on opsonizing and we found no significant results, so I'm just going to exclude that amount of data and go right to duration. So we allowed the macrophages to undergo phagocytosis for either 30 minutes or 60 minutes to begin with, and we found that phagocytosis, the amount of phagocytosed particles each macrophage could consume, actually doubled in the 60-minute amount of duration compared to 30 minutes. So from that time on, we used 60 minutes for that portion. And these are our beautiful pictures that we took. As you can see, the blue is the nucleus, and the red is the actin, and the little green dots are the zymosin, or the phagocytosed particles. So as you can see, in the interferon-gamma, there are many more phagocytosed particles than the unstimulated, which this is what we expected to see. But when we Raft disrupted, you can see significantly less amount of phagocytosed particles. And this also occurred for the non-stimulated. And one other point that I want to point out right now is that these pictures are all to the same scale. So we actually saw that Lipid Raft made the macrophages smaller, too. in morphology, and less spread out, less attachments. Here's a little quantitative analysis. As you can see, Raft disruption in the green caused decreased amount of phagocytosis particles in unstimulated and interferon-gamma stimulated cells. But we saw that in the interferon-gamma cell signaling, or interferon-gamma stimulated cells, that the decrease was almost twofold, like twofold greater than the decrease in the unstimulated. So, going from this, conclusions, target opsonizing does not significantly increase, so it had no great effect on our results. The magnitude of phagocytic index is proportional to the duration, so that 60 minutes had a significantly more phagocytosed particles than 30 minutes. And then we found that lipid raft is in some way involved in the phagocytic ability or functioning of macrophages because of the twofold difference in the interferon stimulated cells compared to the non-stimulated cells. So this lipid rafts do something to affect the interferon-gamma cell signaling pathway. And lastly, as I was saying, the morphology actually changed in lipid raft disrupted cells. They were smaller and less attachment, basically. And then I'm going to hand it off to Luke.
All right, thank you, Kent. As Dr. Doan said, I did the nitric oxide portion, the inflammation, and the specific aims that I looked at were whether raft disruption had an impact on nitric oxide production in the macrophages, and also we wanted to create a quantitative analysis for whether the INOS gene, which is a gene involved, or the cell makes the INOS gene in order to produce nitric oxide, and we wanted to create a quantitative assay for how much INOS was produced. And once we had that assay, we wanted to determine whether raft disruption did affect the macrophage production of the INOS gene. So my experimental approach looked very similar to Kent's. We had the normal medium, the low serum medium, and then the raft disrupted cells, which were also in the lower serum medium. And we subjected it to the same set of stimuli, either IFN, LPS, IFN and LPS together, or no stimulation. And then what I did was a nitric oxide assay, which we used a grease reagent to do, or the INOS gene assay, which we isolated the RNA from the cell, converted it to DNA, and then amplified it to see if that gene was in the cell, or was being produced by the cell. So this is a graph of the nitric oxide production in normal macrophages without their rafts disrupted. You can see IFN-gamma alone, LPS alone, and unstimulated don't really cause the cells to produce that much nitric oxide. But when you put interferon-gamma and nitric oxide together, you see a high nitric oxide response by the cell. And this is the normal medium, and the normal medium cell's response was very similar to the low serum medium. And then we compared those cells to the raft disrupted cells. And as you can see, this is the level that we saw the cells with normal medium produce nitric oxide. And it was significantly reduced when we disrupted the lipid rafts in these cells. So we could conclude that lipid rafts was involved in the nitric oxide production in these cells. So moving on to the INOS data. Down here we have just a control showing that all the cells have the same amount of genetic material in them. And we have the normal medium, low serum medium, and lipid raft with low serum medium down here. And they're either stimulated with interferon-gamma or not stimulated. And you can see the cells that had their lipid rafts intact did produce a significant amount of INOS gene expression. But when we disrupted the lipid rafts, they produced relatively no INOS. So the conclusions were that lipid rafts were involved in the nitric oxide production of these cells, and when the lipid rafts are depleted, there's a significant decrease in nitric oxide production. And also that we showed that lipid rafts were involved in the expression of INOS gene in these cells as well. And I think I'll pass it off to Dr. Doan for the conclusion.
I think I'm hooked back up now. So Luke and Kent both did a fantastic job in the lab this summer, and what particularly intrigued me about their data is that the disruption of lipid rafts appeared to have effects both on baseline macrophage activity, which is not something that I've ever really seen reported in the literature, and it's certainly not something that I have seen working with students in the past. And so that was what Kent sort of saw with the phagocytosis data, where disrupting the lipid raft seems to have an effect on the basic level of cell eating by macrophages, but also a more dramatic effect on the inducible consumption of particles. And then that effect on the inducible consumption of particles was completely consistent with what Luke observed this summer for nitric oxide production, where if you disrupt the rafts, you get less signaling. And that's kind of what we expected based on the fact that we know that the receptors for these stimuli happen to be present in lipid rafts. So all of these data kind of beg the question of what are we doing next? So thinking about the phagocytosis project, there were a few things that I found particularly interesting. One thing that I found particularly interesting was the really dramatic disruption of cell architecture in the presence of raft disruption. You get these cells that are known for doing this and sticking to stuff. So that is my interpretive dance of macrophage structure. Okay? But I mean, they're known for doing that. When cells stick to culture plastic, you can actually enzymatically digest them off. And normally, even for the most sticky cells in culture, you can get them off in 5 to 7 minutes. We tried to digest the macrophages off this summer, like the regular ones under normal conditions. 2 hours. They were still stuck like glue. So the fact that raft disruption causes this rounding and this shrinking and this apparent loss of attachment is really interesting to me, because that probably affects the structures that are holding the cell out, which we would call the cytoskeleton. And so I'm kind of interested in this cell structure thing and lipid rafts. And so you can actually track lipid rafts during phagocytosis, or you can track them just in resting cells using the B subunit of cholera toxin. And that sounds very horrible, that like, ooh, I'm going to put cholera toxin on the cells. This is just a binding piece. It doesn't actually have any biological activity. It just sticks to things. And it sticks to a lipid in the cell membrane that is present in high levels in rafts. And so what we're looking at here is a picture from Invitrogen's website extolling the virtues of one of their products, which can actually stain the lipid rafts. So these are not bone marrow macrophages. They're a different type of cultured macrophages, but all of these little yellow dots are the lipid rafts that are present on the surface of those cells. So it's eminently possible to track these structures by microscopy in the cell types that I work with. And over here, I've replicated one of Kent's phagocytosis images, where you can see these little phagosomes, or these places where the fluorescently labeled yeast have been pulled into the cell. And what you can see in some of them is that they're still very close to the cell surface. Now, if you take away the green and look at just the red, which is the cytoskeleton, what you frequently see is what looks like a honeycomb structure that surrounds every one of those green vesicles. And so those go away, though, when we treat with the lipid rafts. So I'm very interested in, or treat with the raft disrupting agent. So I'm really interested in looking at the rafts to see if they sort of surround these phagosomes in a way, because I think that they might, based on the points of contact. I'm also interested in how disrupting the rafts makes the cells pull away. So that's something that we're looking at for the future. In terms of the project that Luke was working on, we have ample opportunity for further exploration. So I didn't show this in the beginning because I thought it might be a little bit overwhelming, but I think you're ready for it now. So here, what you've been nodding, so I thought I could go there. I wanted to make sure everyone was nodding with me, right? Okay. So we're going to go there. This is the cell membrane. This is the receptor. This is interferon-gamma. So interferon-gamma comes from outside the cell, bumps into the receptor, and what you get in the middle is kind of like a much faster version of, a Pony Express kind of system, or a relay race, where you pass off cellular batons by just little chemical modifications. And so this little orange circle, you don't need to worry about what that is, but you can sort of pretend that that's the baton that's getting passed in a relay race. And it makes information move from up here by the cell surface down here to the nucleus, where all the information is stored for the cell, and it changes the way cells use their information. So what we looked at was the very end product here. We said, if you mess with the receptor and its localization in lipid rafts, what happens to nitric oxide production? And what we found out is that that goes down. The next question Luke was asking is, what happens to the expression of the gene that turns into that protein? And we also found that this goes down. Okay, so now we know that this step is sort of out of whack in Raft-disrupted cells. And so our plan for the future is to keep working our way backwards through this pathway. So if you look at what happens in this pathway, and I'm actually going to do it from the top down, it'll make more sense that way. If you take a look at what happens in this pathway, you see this molecule called STAT1. If you really want to know what that is, I will tell you later. But STAT1 gets a chemical modification, which you can easily detect. So one thing that we could ask is, does Raft disruption interfere with the activating modification on STAT1? My guess is it's going to be affected. Then we can ask the question, well, if we get a reduced level of STAT1, are we also going to shut down other things that are part of the nitric oxide pathway, like this molecule, IRF1? And again, my guess is that that is also going to be knocked down. So those are things that we're looking at testing here. Another thing that's come to my attention recently is that macrophages that tend to lose these capabilities often gain alternate capabilities. And so we don't really have time to talk about those, but it's called an alternative phenotype. And so I have made some contacts to develop protocols to start looking at alternative macrophage phenotypes in the future. And it looks pretty easy, which is exciting. So finally, some acknowledgments. I would, of course, like to thank Deb's office and the Edgar Scholars Program for graciously funding my 2 students. We had a great time this summer. I'd also like to thank the Faculty Development Grant Program, because they graciously funded a number of the more expensive reagents that we had to purchase for the summer, as well as the Department of Biological Sciences and Science Division, which does provide funding for all student research. Dr. Brian Hyatt needs to be acknowledged for his contribution to our gene analysis of INOS. He walked both of us through the practice of doing these quantitative analyses. And we also need to thank Ms. Renata Thielen, who is also a College of Arts and Sciences biology senior, who was instrumental together with Kent in putting together the phagocytosis assay. Kent worked out a lot of the conditions. Renata gets a little obsessive about visual representations of things, and so has gone very far in terms of imaging and has taught me some things about how to use the microscope. So we can't leave her unacknowledged. So that's where we wrap things up, and I think we still have a few minutes for questions. Yes, Ken.
Well, it's sort of trying to think about what is the implication of, so you mentioned something that the loss of lipid rafts means sort of a cell shrinkage. Mm-hmm. And so can you talk a little bit more about what might be the implications of that, or how might we sort of, how might they either be useful, or what information might you be able to find, or what is the implication for the body or the cell that happens?
So that's a really interesting question. So we didn't have time to talk about, I worked with another student last summer, Melissa Bellick, who got some really interesting data from what are called transformed cells. So these are essentially cancerous macrophages. And if we compare the experiments that Luke did this summer with the experiments that Melissa did last summer, what we found is that in normal cells, because the bone marrow cells are normal, we call them primary cells, Raft disruption completely knocks down what's going on with cell activation. In the transformed cells, activation goes up on a per cell basis, which is really interesting. And so I'm thinking that, typically when you see things like loss of attachment sort of coordinates with a metastatic phenotype in cancers, for example. increased inflammation is not a good thing. And so one of the things that I'm interested in possibly looking into for the future is doing Raft disruption, not by this more traditional quick method, but using a slow method that involves growing the cells in the presence of cholesterol-lowering statin drugs. And so basically you stop the cells from producing cholesterol, and then you grow them in medium that won't let them replenish it. And I'm kind of interested in comparing the cancerous cells versus the primary cells, because if the cancerous cells actually become more activated and more inflammatory, that could have some interesting implications for the use of those cholesterol-lowering drugs, which I want to say more than 20 million Americans currently take, and that number is expected to double and extend into other age demographics over the next 10 to 20 years. So does that answer your question?
Yes.
Yeah. A lot of the other questions are just sort of very pure cell biology, thinking about how do you alter signaling, because lipopolysaccharide is, in experimental models, you can't use it in humans, but it's you can use it as what's called an adjuvant, which is something that makes vaccines work better. And interferon-gamma is sometimes used as an immunotherapy for certain viruses and potentially in cancer systems. And so thinking about how those signals get regulated can also be very important for thinking about structural regulation of signaling in medical treatments and applications.
I have a question, but I don't know how to ask it. So the macrophage that eats, does it take on both bacterial and viral things?
So what all does the macrophage eat is kind of the question. It will consume anything that's large enough to recognize. So it will eat bacteria, it will eat clumps of viruses, it will eat fungal cells, it might eat small parasitic cells, it will remove damaged, or dead red blood cells from the circulation, damaged cells from other places. And then, so my anecdotal fun fact about macrophages is if you know anyone with a tattoo, when they inject the tattooing, the macrophages that underlie the skin actually ingest the dye, but they can't digest it, and so it gets stuck there. And then as those cells die and are replaced, new cells come along and consume the old cells, like, can't digest the dye. And so over time, that's why you see a little bit of fading and migration, is if you get incomplete consumption of the previous cells.
So tattoo research.
So, well, I'm not planning to do tattoo research, but I once had a very well-respected scientist tell me that that's why tattoos stay where they are, is because macrophages ingest the dye. So they'll eat just about anything that is foreign and particulate. Yeah.
Can I ask a student's question?
Sure.
For both of you, I'm just really interested in what surprised you about the research that you did, or the research process that you really spent a whole summer doing this research. Was there anything particularly surprising that stands out to you? And then what's next for you? What are you thinking about after that?
Well, I can. The most surprising thing is, for me, because the phagocytosis assay took like 2 to 3 hours, and if you made any little mistake, any part along the way, the whole, all your data was screwed up, basically. So you had to be very focused on little details all along the way.
And Kent also learned a lot about incremental progress. So it's like, do we opsonize? How long do we do it? What concentration do we do it with? Do we try it again? And so, it probably took over half the summer just to develop the assay. So a lot of his work went into development.
Yeah, I would say a lot of my work also went into development, especially with the gene expression. It took a lot of trial and error to get the results that we wanted to see. And yeah, it was interesting to see how much can go wrong, I guess, and what you have to do, or how much you can learn by that, I guess, and in adjusting it and researching more and learning more about it. So, I thought that was...
Do you remember my big motto? If you only had to do it once, they just call it search.
So what's next in your lives?
I am applying to medical school, so I think kind of the same. So I think research is really helpful in understanding more about immunology and appreciating all that more. So